Welcome to another video tutorial. In this video, I will show you an overview of how we can interpret the results of microbiome diversity analysis. In this video, I will show you the website by which we have analyzed our data, and I will also show that how we can interpret the results which can be used in our research articles. So let me go to that website. So my website is majorbio.com. My website is in Chinese. So if your website is also in Chinese, so you can go here and you can translate into English version. So if this fails, you can simply just go here and you can also click on this translate to English as well. So you can see now the website is translated to English and you can log in with your own login and password. So here you can see my login and password is already saved. You can use your own W. So you can click on this login. So you can see here now our website is just login, and you can see here it is showing the overview of our current analysis report. So you can simply click on this view report. It will show you your recent analysis which you have just performed from that company. So you can click on this view report. It will take you to a new screen. So we can translate again it into English by using this option. We have our report which is ready, and here we have performed 14 analysis. Any microbiome analysis, the overall flow of data is the OTU analysis, and OTU has pan analysis, pan genome analysis, and core genome analysis. Then we have alpha diversity and beta diversity. Alpha diversity is further divided into dilution curve and community bar diagram and pie diagram. Similarly, we have the further division of beta diversity. And in beta diversity here, you can see we have PCA, that is principal component analysis. And similarly, we have beta diversity principal coordinate analysis. And later on, we have NMDS analysis. While later on in the video, I will tell you that what is NMDS analysis. And at the last, we have species composition analysis and sample to species relationship. And we know that we usually calculate it the Sirkos plots. So before we start the farmer analysis of OTU and alpha and beta diversity, so first we should have information about the samples. So to get the information about the samples, you can simply click on this data information management, and you can click on this first sample information management. So when you will click on this, you can see here the information of sample sheet is present. You can see here we have 3 technical replicates of control negative, and similarly we have 3 technical replicates for control positive, and we have 3 technical repeats for management. Actually in this design, here we have applied one disease. So in this control negative, we did not apply any disease because this is normal. And in this control positive, we have applied one disease. And here to cure that disease, we have applied medicine, and we have just compared the microbiome diversity. And in follow-up one by one analysis of OTU, alpha and beta diversity, I will show you what our results are telling. We can go back here and we can click on this first OTU analysis. So before I go into the detail, let me show you that what is OTU. Actually, OTU is a set of sequences which are closely related together. So if I can show you that what is OTU, so if we largely talk about first, we know that overall relationship, the largest is the domain. Then domain is divided into kingdom, and kingdom is further divided into phylum, and phylum is divided into class, and class later on into order, and order is further subdivided into different families, and families are divided into genus and species. Taxonomic units of microbiome diversity analysis. So I can show you the definition of OTU. So you can see OTU stand for operational taxonomic units, and this is the set of closely related sequences. If we go back, so here is our first, the pan analysis. We should know that what is pan analysis. You can see here on the x-axis we have number of samples, and we know that first three is actually control negative, and the next three are control positive, and here next three are the management group. And here if we see the control negative has lowest number of OTU. OTU mean operational taxonomic units, which is referring to the sequences. So it means actually the control negative has lowest number of closely related sequences. But when we talk about control positive, you can see the number of operational taxonomic units which were associated with one another increases. And if we talk about the management group, we can see the number of operational taxonomic units are increasing while we deal with the management group. So here you can see this OTU is further divided into two. One is pan genome analysis, and further is the core genome analysis. So first we should know that what is pan genome, or what is the pan analysis. Pan genome is actually the set of whole genes within a clade, is called pan genome. And what will show the pan analysis, actually it means it is showing the diversity of all strains in within an enclave is called pan analysis. So similarly, what would be the core genome? So core genome would be the only set of those genes which are common between the different samples. So if I click to the core genome, you can see here now it is, you can see here the core analysis is actually showing the opposite trend with respect to the pan genome analysis. You can see in the first control negative, the first three sample is showing the decrease of shared OTU or shared diversity between the different samples. And if we see between the control positive, you can see the number of shared or the common OTU is further decreased. And if we see further in management group, the overall shared diversity or shared strains between these samples is further decreased. So it means our first analysis of OTU is showing that in the pan genome analysis, our first control group has highest pan genome, but it has lowest core genome. So now, in order to further understand, we have to divide this pan genome OTU and core genome OTU into further alpha diversity and beta diversity. So first, let's try to go into detail of alpha diversity, and we can click on this estimation. You can see here now the results of our alpha diversity are uploading. Here you can see here we have sample name on the x-axis, and similarly we have here S index on OTU level. So if we see further, here are the different indexes. So we should know that which type of these indexes. These are actually the alpha diversity indexes. And here the most famous one are these Shannon alpha diversity index and Simpson alpha diversity index. And here are all other alpha diversity indexes are based on the relative abundance. But here this Shannon is based on, here I can show you the detail. You can see that the Shannon index is based on the richness, and similarly the second one, SIMP index, is based on evenness. So what is actually the richness and what is evenness? So you can see here the richness is showing the number of species within ecosystem. So if we have to strength on the richness, it means, let's suppose we have two species in which the control negative have three species, and similarly control positive has three species. So you can see now the control negative and control positive has both three number of species. So it means the overall richness of these control negative and control positive is same. But sometime we know that control negative although the number is 3, but maybe it has a different kind of bacteria or different kind of microbiomes, then always the richness will not work for us. Then we have to go with the evenness. So evenness can also refers to the extent that which species are evenly distributed, and it means which proportion is making that number. So it means when we have to focus on the richness, then we use Shannon index, or when we need to give weightage to the evenness, then we use Simpson alpha diversity index. And other all diversity indexes are only the relative abundance based indexes. So here you can see in the control negative, it is showing increasing and decreasing trend. It might be due to the technical replicates difference. Here you can see the control positive, these two has equal, and similarly the management group has lowest number of S indexes alpha diversity. So it means uh, if we take alpha diversity estimators according to the S index, you can see the management group has lowest alpha diversity index. So we can also modify this chart according to our need using these integrated tools. You can see here we have chart tools. Similarly, we have deposit report, instruction for use. And similarly, if we want to download this figure and want to use it as such in our article, so we can here uh, change the format. Like if you want to download in the form of TIF or PDF, so you can download and use it. Similarly, if you think that this title need to change, so simply you can double click on this, so you can change this according to your own. You can first click on this edit text, and similarly you can double click on this and you can change this text according to your need. So we will move next to the dilution curve. If we double click on this, so it will show you the dilution curve, diffraction depth at all. So here it is showing the diffraction curves, and here you can see it is showing the SOBS index on OTU level, and here it is showing the number of reads. So why we do this? This is actually a part of quality control system. I mean, whenever we perform our analysis, we have to perform certain quality parameter analysis to check whether our analysis meets the requirement to take it to further studies or not. We don't need to focus much on this analysis. I will make a separate video on this so we can move further to our community bar plot. So you can see here our community bar diagram and pie diagram is divided at two level. One is at the phylum level and one is at the genus level. So if we click on this community bar pie at phylum level, you can see that our results are updating. So here you can see our community bar plot analysis is now loaded, and you can see on the y-axis it is showing the percentage of community abundance on the phylum level. And if we see if we mouse over any color, so it is showing, you can see here the red color is showing the firmicutes. So in control negative, the most of the abundance is related to the firmicutes, and here if we mouse over to this one, it is showing the protobacteria. So it means this control negative 2 has highest abundance of protobacteria, which is probably very different to these two. So it might be due to the technical replicate difference. So we have to only focus on this one. So if we take to the management group, you can see this path which was showing to the protobacteria is minimum as compared to all these two. So we can say that community bar plot is showing that when we have applied the medicine to control the disease, it has probably decreased the quantity of and diversity of protobacteria. So this way you can interpret the results. And if we see the community bar at genus level, you can see it is showing the percentage of community abundance on genus level, and here are our sample name, and here you can see here we have the different genus level, and if we mouse over to this, you can see it is further showing the another type, that is Limocylactobacillus. So the overall quantity of Limocylactobacillus has decreased when we give disease to our animals. Similarly, if we further see that this color is showing to the Limocylactobacillus, if we mouse over to this Klebsiella, it is only present in the control negative. So if we mouse over to this one, that is Novosphygnobium, so here you can see in the management group, it is showing the least quantity. Similarly, here the quantity of Enterococcus is also decreased in the management group. Similarly, this is all only present in the control negative. And if we take only the Streptococcus, you can see this is only present in the control negative. So it means overall the quantity of all these while moving from control positive to the management group, the overall quantity of all these genus level microbes is decreasing. So similarly, now we can move to our next community heat map analysis. So you can see here we have community heat map analysis. So here I have performed another one, which I have just used my own. So I will only show you this community heat map, which is showing heat map at all. So here you can see it is showing the community heat map analysis on the genus level. It is just showing the random. It is just showing the random arrangements of our sample. Although we know that control positive should be together, control negative should be together, and similarly the management group should be together. So to edit this chart, you can simply go here and you can design your own. Here you can see this is the grouping scheme. Let's suppose you only want to compare control negative, control negative, control negative with control positive, 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 and you can just run this. So after you will run this, you can see now it is being calculated. Please pay attention to the analysis record. So you can see here now our one analysis is being calculated. So it will take a few seconds to load our results, but meanwhile I can show you and that how we can interpret these heat map analysis. Here actually in this heat map, each color is showing one genus. So it is important to note only those genus in your favorite sample. You can see here, let's suppose if someone is red in your three technical replicates, but it is showing other color like blue. So it means if it is red colors in one sample and blue color is in the second sample, so that could be your target genus to study further or to indicate its results. So similarly here you can see if we talk about control negative, this is control negative 2. Similarly, this is control negative 1 and this is control negative 3. So here is red, and here is red. But if we talk about in the control positive, so here is blue and here is blue, and where is control positive? Here is also blue. So it means the staphylococcus are showing the differential abundance at genus level. So now we will move to the beta diversity analysis. We should understand that what is actually the alpha diversity analysis and beta diversity analysis. If we need to understand only the diversity within the sample, like if you are only considering the diversity within control negative, so this is actually the alpha diversity. But if you are comparing the control negative one diversity with others, so that would be called the beta diversity. So it means if you are comparing within sample, like you are comparing only looking that what are present within this control negative, so it would be called as alpha diversity. But if you are comparing the diversity present in control 1 and comparing it with control positive 1, so it would be come under the heading of beta diversity. So you can see here, previously I have shown that which one was calculating. Now it has just been uploaded and finished, and we know that it was actually the heat map, and here it is showing in the green color. So you can see here our customized community heat map is being updated, and here you can see we have only now a fine comparison of control negative and control positive. Similarly, you can also design your own community heat map and you can arrange them according to your need. We will move to now the beta diversity. As I have already told, in beta diversity we actually compare the diversity within different between different sample. You can see this dendrogram is showing a further. You can see this one is major category and this one is in under one clade. This is in one clade because here is one major branch and one major branch is further divided into this. You can see here this control negative 2 and control negative 3 was under one clade. So it means they are more closely linked. And similarly, if we see management group and control positive group, they are more closely linked. So here is the distance of that branch lengths. And if we mouse over to this one, and we can see we have the different operational taxonomic units 1. And you can see overall its quantity of OTU21 is increased in the medicine group and control positive group. Just as a quick note, I want to mention that you can see here, did you feel an odd thing in this analysis? You can see here the control negative 2 and control negative 3. They are majorly under one clade. But if we find control negative, it is showing a very different clade. So probably while performing experiment, the students have done a sampling mistake, and you can see here probably they name control positive 3. They should name control positive 3 as a control negative 3 because all these control negative 3 should be in one group, and similarly the control positive should be in one group. So but here you can see the control positive 3 is overlapping with these two, and probably this is a sampling mistake. Anyhow, our concern in this video is only to understand the trend. So let's move ahead. So now we can move to next, beta diversity PCA. So we know that PCA stands for principal component analysis. And here you can see the first PC1 is showing the variation of 30% and PC2 is showing 25%. So overall, it means if we perform the PC analysis, so the overall diversity through PC1 is 30% and at PC2 is 25%. So it means overall PC1 and PC2 is making, if we add them 30% and 20%, it means overall 55%. If we take the beta diversity at principal coordinate analysis, PC principal coordinate analysis is similar to that principal component analysis. And here you can see now our samples are more easier to identify. You can see here on the sample CP3, this is sample CN3, and this is sample CN1. So it means the control negative and control positive are more linked together. So it means because they are placed closely together, so it means they are more closely linked together. So similarly, you can see here management group. This is also management group. So it means the management group are also placed together. So it means our principal coordinate analysis at PC1 is 56% and PC2 is showing 30% variation. So the lastly, we have beta diversity, and usually we perform NMDS analysis. We usually perform NMDS analysis to show the relationship at OTU level. And here you can see this is the NMDS1 and NMDS2. So why we perform NMDS? So here I can show you this NMDS is actually the non-metric multi-dimensional scaling test. Although to give you an easy understanding, you can see it is hard to, let's suppose, because here this is y-axis, this is x-axis, and let's suppose your one sample is placed here, your second sample is placed here, and there would be too many dimensions. So our purpose to for this NMDS analysis just to reduce the number of dimensions, because if we take x, y, z and many, so it would be hard to draw on 2D surface. So just to draw it on the 2D surface, we perform this uh, non-metric multi-dimensional scaling test. So overall purpose would be only the data analysis method that reduces the research objects. It means here our samples are our objects, and that is just reducing the multi-dimensional space to the low dimensional space. Here are the circos plot, and you can see here we have management group, and here we have control positive group, and here we have control negative group. So to understand further, you can see the overall, see in the control negative, you can see we have protobacteria 7.4% and actinobacteria 0.39%. But if we take it to the control negative 2, so now the protobacteria is 55%, and similarly in the control negative 3, protobacteria is 3.8%, and similarly the control positive, it's, you can see now the protobacteria overall concentration diversity is decreasing, that is only 0.35%, and similarly you can see here is 0.68%. So it means the overall protobacteria in control positive is decreasing. So if we try to associate this, so you can see it is associating with the firmicutes. So in the blue color, you can see if we take this blue color, it is showing to the different this color, and it is showing to the association with another species, that is the firmicute. So it means it will increase the quantity of firmicutes. So similarly, if we take to the medicine group, you can see here in the medicine group, the protobacteria is again high, 6.3%. But in comparison to the control negative, it is still low. But in comparison to the control positive, it is more. So if we try to associate the relationships, you can see it has high relationship with the firmicutes as well. And it is showing also a linear relationship with all these like actinobacteria, cyanobacteria, and bacteroids and others. So in this way, you can draw your relationships, and to get further, you can see here. Here, the length of the sample refers to the relative abundance. Like highest abundance is in this red color, and red color is showing to the protobacteria. So in this way, you can have a rough interpretation of your microbiome data. To get further detail, I will make a separate video of each one. So if you like the video and or have any question, please let me know in the comment section. Thank you for watching. Bye bye.